Alright, this is your cellular respiration pre-lab video. Cellular respiration is, of course, referring to the process of oxidizing glucose in order to make a usable form of energy. Okay, so we're going to take, for example, we have glucose, which is C6H12O6 plus O2, forming CO2 plus water and O2 and energy. Okay, now if we were to have our equation balanced, of course, we would have six oxygens there, six CO2s there, six waters there, and then the energy is mainly referring to ATP. Okay, this process happening in the mitochondria of both plants, animals, and other eukaryotic cells, and then even though the production of ATP is happening in prokaryotic cells, it's not happening in the mitochondria since prokaryotic cells don't have mitochondria. All right, so um, what's going to be happening here, this is, again, what we call the phosphorylation or the oxidative phosphorylation of ADP to form ATP, which is ADP plus PI is going to form ATP, and this, of course, is our usable form of energy that organisms use for their daily anabolic processes. Now, in this lab, what you're going to be doing is we're going to be taking a look at black-eyed peas. Okay? Now, these black-eyed peas, okay, are obviously plants. And these plant cells are going to undergo respiration at different temperatures, and we're going to measure the respiration rate. Okay? And this is how we're going to do it. We're going to have 1, 2, 3, 4 different tubes, okay? And in these tubes, I'm going to have some germinating black-eyed peas at room temperature, which is about 25 degrees Celsius. I'm going to have some germinating peas at 50 degrees Celsius, so a little warmer. And then I'm also going to have some germinating peas at 50 degrees, not 50, 0 degrees Celsius. Okay, and then lastly, I'm going to have some non-germinating peas, and those will be at room temp. Okay, so about 25 degrees Celsius. Now, the difference between the germinated peas and the non-germinated peas is that the germinated peas have been sitting in water for the past 3 days, so they have undergone the process of starting to want to, grow a new plant. However, the non-germinating peas or beans have other peas we're just sitting in the bag just before this lab. So what you're going to be doing is you're going to be measuring the pressure inside the tubes. Okay, so we're going to have these tubes sealed off, right, with the peas inside them. And what's going to happen, obviously, is if cellular respiration is going on, we're going to be producing carbon dioxide and then going to be using up oxygen in order to make that ATP for the plant. Now you're wondering, well, if we look back at our equation, I am producing, right, 6 CO2s for every 6 oxygen. How is my pressure going to be affected? Well, what we're going to do is we're going to have a chemical reaction taking place that's going to absorb all of the carbon dioxide that is in the tube, and because all the carbon dioxide is going in the tube, the only thing that's going to be happening is a change in oxygen pressure, okay, or the partial pressure of the oxygen. Now, in this process, okay, the oxygen should be consumed because we're using that oxygen to oxidize the glucose in order to make the ATP. Okay, so any pressure changes that you're noticing inside the tube are going to be a result of the decrease of oxygen or the oxygen consumption. And eventually we're going to get the rate of oxygen consumption based on these different temperatures. So now let's take a look at a sample tube and kind of get an idea as to what you're going to be doing. Okay, one, you're going to have just a normal test tube. Okay, now on the bottom of the test tube, we're going to have some absorbent cotton, some cotton ball. Okay, now in that cotton ball, I'm going to have some potassium hydroxide. Well, as it turns out, the potassium hydroxide will react with the carbon dioxide. Okay, so if I was to say, oh, here's the carbon dioxide, so the potassium hydroxide and the carbon dioxide is going to form the potassium carbonate and some water. Okay, so this is how we are going to be absorbing the carbon dioxide inside the tube. Okay, so this is what's going to cancel out any carbon dioxide that's being respired from the plant, from the beans or peas. Okay, now the next little layer right here is going to be some non-absorbent cotton. Now, the purpose of this non-absorbent cotton is just to separate the potassium hydroxide, strong base soaked cotton from the beans that are going to be right here, or the peas that are going to be right here. Okay? Right? Now, there's going to need to be a certain volume, okay, a specific volume. Okay, specific, oops, forgot a word there. Specific volume of the peas. Okay. Now this is important because obviously, if we remember back from chemistry, if we remember Boyle's law, if I have a varying, because remember you're going to have multiple tubes here. If I have varying volumes of space inside the tube, that's going to directly affect the pressure. And since we're measuring pressure, we need to make sure that we have the same volume of substance, in this case peas, in this section right here. Okay, so we'll have the volume of peas, and then lastly we're going to have a little stopper, which is going to fit a little better than this drawing that I have. Okay, and inside that little stopper there's going to be a sensor, and this sensor is going to be connected to one of the interfaces that's going to measure the pressure. Okay, and the pressure is going to be in kilopascals. All right. So this is going to be your main setup for each tube. Okay. Minus one thing, we need to make sure that we also regulate the temperature inside this tube. Okay. Now, when I say you're going to have different tubes at, 50 degrees Celsius, 25 degrees Celsius, and 0 degrees Celsius, understand that to better control that temperature, we're going to have this tube submerged in water. So you're actually going to have an Erlenmeyer flask full of water, and then the tube is going to go in it, like so. So we're going to have, here's the opening of the flask. Not horrible symmetry, but and then of course there's going to be water inside here as high up as you can go. Okay. Now in this picture right here, I have the stoppers hanging outside of the flask. In truth, the flask will be all the way up here. Okay, or wrong color, I guess. Here. So the flask really is going to be pretty much submerging the entire glass portion. Now, if I was to zoom in on this section right here, okay, we're going to have the edge of the tube, and then the stopper, right? Well, the tube will be right here, right? The test tube. I don't want the water to be higher than this portion right here. You want to get as much of the tube under the water as you can, okay? Because we really have to regulate the temperature of the air that's inside this tube, and of course the water there will help do that. Okay. So a couple things we have to regulate. One is going to make sure that we have equal volume of our sample inside there, because that will affect the pressure. And then again, we'll submerge it in water so we can also regulate the temperature, and that will also help regulate or help give more accurate results for our pressure. So we just answered the question on how we're regulating temperature, but I really haven't addressed how we're regulating the volume. Okay, because just as it turns out, a germinating pea is roughly that size, and a non-germinating pea is smaller. Okay, they don't take up the same amount of space. Now, in your different tubes, your germinating versus your non-germinating, and all of your all of your germinating tubes, you're want to have an equal number of beans, because obviously, if I have, 10 of these, but I have, no, let's go ahead and say I have two germinating peas. I have 10 of these germinating peas at 25 degrees Celsius, but I have, 50, which there's no way we're going to fit 50 in that tube, but say there's 50 in there at 50 degrees Celsius. This right here is going to be an issue, okay? Because obviously 50 peas is going to be respiring and absorbing more oxygen than only 10. Okay, so we need to make sure that we have equal number of beans or peas, right, in each tube. Okay, so that's one thing we need to have. We need to have equal volume as well. Now, like I said, because you have two different types of peas, right? One pea is big and one pea is small. How are we going to compensate for the smaller pea? And we're going to do that by using glass beads. Okay? So it really, if I was to take a look at what's going to be in, say, the two tubes at 25 degrees Celsius, the germinating versus non-germinating, this may be 10 peas, and I haven't decided on exactly how many peas I want to add to the tube yet, and we'll take a look at that when we get to class. Okay? And let's say I'm going to have 10 of these guys right here, but this may only be, or this may be 7 milliliters or 7 cubic centimeters worth of volume, but this is going to be 5 milliliters or cubic centimeters of volume. Well, that's a problem. They're not equal volume. So what you'll do is you'll be adding, for example, 2 milliliters or cubic centimeters worth of these beads, and that will regulate the volume. And since the volume would be the same and the number of beans would be the same, then that's just one less variable we have to regulate in the experiment. All right, so to recap, you are going to have 4 different tubes. In each tube, you're going to have some germinating peas or non-germinating peas. Okay, so 3 of the tubes, right, will have germinating peas, and they should again be at an equal number of peas. Also, there should be equal volume. Okay, and if say 20 in these 3 tubes, let's say if we had 10, 10, and 10, if those 10 end up having a greater volume in the 25 degrees Celsius tube than in the 50, then of course we're going to add glass beads to compensate for that volume. You're also going to have one tube that's going to have non-germinating peas. Okay, and in both of these tubes, you're going to run, or sorry, in all 4 of these tubes, okay, you're going to run the experiment, okay, for 15 minutes. Okay, so the experiment itself will run for 15 minutes. You do not need to keep track of time. The interface, the lab quest will do that for us. Okay, but the one thing that we need to do in order to make sure that these 15 minutes count is we need to have about a 10-minute calibration period. Okay, so calibration, and then this is actual the trial. Okay, so your trial is going to be 15 minutes. Your calibration is going to be 10 minutes. All right. Now, obviously, if we were to do the math and knowing that I don't have that many interfaces, this lab is actually going to be split up into a couple days. Okay, so you may do, or some groups may do this lab on the first day, and other groups may do this lab on the second day, just to make sure that everybody gets it done and that we have enough interfaces to do this. Okay, but we have 3 tubes of germinating peas at different temperatures, and then we have 1 tube of non-germinating peas at room temperature. For all of these tubes, before we start the trial, we must have a 10-minute calibration period so the pressure can even out before we start, and it gives us time to make sure in that 10 minutes to really make sure that your temperature is remaining constant inside the Erlenmeyer flask. And then once you're ready to go ahead and start your trial, and it'll go for 15 minutes. Okay. There's no reason why you can't do more than 1 tube at one time. In fact, I encourage that. If you do, say, 2 at a time, then that will allow you to, while you're calibrating one, you could be setting up the other one, and then once the calibration for the first set is done, then you can start the second set, or run the second set, and then while that second set is run, then of course you'll have the second one calibrated and ready to go. Okay? So if you guys have any questions, please make sure you ask, and make sure you answer the questions below, because I really need you to be able to understand what's going on in this lab before you get here, because time's going to be short.